Hi guys! Welcome to episode 3 of our Biochemistry at Home. I am your host for this week's episode, Louis Christian Domingo. So guys, pagpasensya nyo na ako ngayon dahil sa mask ko ngayon kasi inubo ako ngayon, so hindi maganda yung pakiramdam ko ngayon. But I'll still continue to be your host for this week's episode, and I'll be showing to you our experiment for today, which is all about the DNA. So ano nga ba ang DNA? Madaming nagsasabi sa atin na ang DNA is called the blueprint of life. Bakit? Dahil kung wala ang DNA, walang living organisms, anong muha dito sa Earth ngayon? Dahil sa DNA, dun naka-store lahat ng functions, lahat ng traits na needed for an organism to live, for growth, development, and etc. Ngayon, I'll be showing to you the materials needed and the procedures for the experiment that we will conduct today. So, see you guys later. Bye!
Materials needed for the experiment: a cup of water, dishwashing liquid, salt, papaya, tissues, alcohol, clear plastic, and strainer. First, add 1 tablespoon of salt into the cup of water. Next, add 2 tablespoons of dishwashing liquid into the salt solution. After adding them, mix well. Next, pour the solution into the papaya. After pouring it, set aside. Now, filter the solution using a strainer. Filter it again using tissue this time. Lastly, pour alcohol into the solution. Now wait until white particles precipitate into the alcohol on top of the solution.
So guys, now I will present to you the person who will discuss to us the happenings on how the DNA extraction happened. Let me present to you Janelle Fiatco.
Hi, I will be explaining to you how we ended up in getting the DNA of papaya. First, we have to know how the materials contributed in extracting the DNA of papaya. Dishwashing liquids release DNA from the cell by pulling apart the fats and proteins that make up the membrane surrounding the cell and nucleus. In salt, positively charged sodium ions will shield the negatively charged phosphate groups of DNA molecules, helping them to precipitate out of the solution. With the solution that contains water, salt, dishwashing liquid, it degrades the cell walls and membranes, further permitting the release of DNA. After blending, it is necessary to filter the mixture with the use of cloth or filter paper to separate the nucleic acid from the remains of the cellular membranes. Alcohol causes the DNA to precipitate. When DNA comes out of solution, it tends to clump together, which makes it visible. Pouring the solution into the ethanol will make the DNA molecules precipitate into the upper layer of the alcohol.
Thank you guys so much for watching episode 3 of our Biochemistry at Home series. Now, tune in into next week's video as we talk about proteins and more. Thank you guys so much. Bye!